A very big hello to all my wonderful KCET aspirants. I hope you're doing really great, my dear children. And I welcome you all to Masterstroke series, where today we are going to complete biotechnology principles and processes in one go. Now I know this chapter is kind of different from the other chapters of biology. It's actually a whole different field that is biotechnology. And I'll tell you, it's very much interesting. It's just that in your NCERT, the things are given here and there to just make you confused. But trust me, this chapter is very interesting and very easy. You would love this, okay? So basically, in this one shot, I am going to make you fall in love with this particular chapter. But before that, I just want one thing from you. Take out your NCERT, have a pen in your hand. Whenever I say that this type of questions could also come and you think that, oh, I never expected that. So basically, you can just write it down. Okay, this is the type of question that could be framed in KCET and things like that. Works? So shall we start? Yes. So over here, let's start with the first thing, that is steps involved in recombinant DNA technology. Since, as I said, it's totally distorted in your NCERT, I thought I'll make a, structured way of learning this particular chapter. So first thing is that you should know what is recombinant DNA technology. Recombinant DNA technology means I have, for example, my DNA. Okay, my DNA is the gene of interest. My DNA is very much important over here. Now, I'm going to insert my DNA into some other DNA strand as well. Okay, some other DNA strand which has a site for replication. So, the whole thing will be replicating, including my gene of interest which is there. Okay. Now, this new combination of DNA which is there is known as recombinant DNA. What do you call it? Recombinant DNA, my dear children. And for the formation of this, whatever steps are involved are being put up over here. Cool? So, why is it actually used? For example, in my body, there is one gene that codes for some protein. And I want to increase the production of this particular gene. Of course, I cannot make my body do that. I cannot tell my body to do that. We do not have mechanisms for that. So, what do is that we take that gene, okay, we take that gene and then put it in some other organism which basically can replicate this particular gene of interest. And when you're saying that DNA is getting replicated, that means whatever protein it is going to make, it is also going to be made in very much amount. You get it? So that is why we are going to use recombinant DNA technology. So the first thing that we need is isolation of genetic material. For example, my body itself. Okay, in my body, my cell, what do we need? We need the DNA. But in my cell, there is RNA, there is protein, there are a lot of things. So I have to isolate the genetic material. That is the first thing. Second thing is that after isolating, I have to make sure that I do not need the whole strand of DNA. I just need a particular gene which will be coding for one particular protein, right? So I have to use an enzyme that is known as restriction enzyme, my dear children. Restriction enzymes are very particular with where are they going to cut, okay? So these are the ones, they are also known as molecular scissors, and they are going to make the cut over here. Suppose they made a cut over here, right here, as you can see. So this was the actual DNA, they made a cut and now I have this particular segment of DNA. Now what happens is that basically you see there are different fragments. There are different fragments. You cannot just pick directly one fragment over there. You do not know. Okay, you do not know, they are small molecules, right? So what do? You have to separate this particular fragments. You have to choose a smaller one, which is your gene of interest. So what do? You take the help of gel electrophoresis method. In gel electrophoresis, in a chamber, the DNA fragments are being put. Okay, and DNA is negatively charged, so it will be moving towards the positive side. Okay, so it will be moving to the positive side, and the ones which are smaller, they will be moving really fast. The one that is large stays there. So the smaller ones move really fast, and I can just pick them. Okay, this is one I pick them. And now what do? One is not enough for me. Basically, I cannot cut my genes from my body. So what do is that one gene that I have, I am going to make multiple copies of it. And this process is done with the help of something that is known as PCR, polymerase chain reaction. And this process is known as amplification of DNA. You are amplifying the amount of DNA which is there. That is done. Cool, that is done. After that, what do? So you have the gene of interest which is there in very much amount. Now you are going to finally put this particular gene of interest into something that has the ability to replicate, that has the ability to make multiple copies. And what is that something? Usually it is a plasmid. Usually it is a plasmid, my dear children. And plasmid, what are those things, my dear children? Suppose you have Escherichia coli, which is a bacteria. Now bacteria will have its own chromosomal DNA that will be coding for the proteins that the particular bacteria needs. But apart from that, in the prokaryotes, there is some extra chromosomal DNA that is not going to code for any protein or anything like that. They're just there. They have some site which is known as origin of replication, and whenever there is a site that is origin of replication, this is where the DNA polymerase will come and attach and the replication will be starting. So you know one more thing, that is this guy is able to replicate itself. It is able to make copies of itself, and it does not have any link with the proteins that are made up in this particular E. coli. So even if I remove it, even if I add it, there is nothing that is going to happen over here, until and unless I am putting some genes over here which would be coding for certain proteins. So what I am going to do is, I am going to take this particular plasmid. Okay, I will call this as a vector because it is going to carry my gene of interest. So I am going to take this particular plasmid. I am going to make a cut over here with the same restriction enzyme that I made cut over here. Okay, so I will tell you why and all later. So I took the restriction enzyme, I made a cut over here. Now I am going to take the gene of interest and put it over here. And I am going to ligate it, make it together with the help of a very important enzyme which is known as DNA ligase. So DNA ligase will be sticking both the ends over there, my dear children. Now this particular plasmid which is there, my dear children, I can call it recombinant plasmid. What will I call it? Recombinant plasmid. It has my gene of interest. Now the cells which are going to take up this plasmid, okay, the cell, suppose I have a plasmid like this and I have another plasmid like this which does not have my gene of interest. Suppose both the plasmids are kept over here and both of them are taken by the cells. Both of them are taken by the Escherichia coli. I'll just say that the cell has underwent transformation, it has taken up something. Now, how do I be sure whether this one is the one that has my gene of interest or is the one that does not have my gene of interest? Basically, I have to check for it, and this is basically checking for recombinants. So the first thing that you check is whether they are transformed or not, whether they have taken up the plasmid or not. Okay, any plasmid which is there. Now, the second thing is that whether they have actually got the recombinant one or not. That is checking for the recombinants. So suppose it has taken up the recombinant, I will now call it a recombinant cell. I will now call it what? A recombinant cell. Once I am sure that it has my recombinant DNA, my dear children, what I am going to do is I am going to make sure that I am going to do the large scale production of this particular cells. So I am going to make these cells increase their number tenfolds in bioreactors and stuff like that. Okay, so expression of foreign gene is something that I missed over here. So it's nothing but basically I am making sure that this particular E. coli or whatever organism is there is basically producing the protein that I am looking forward for. And once I am sure about it, then I am going to do large scale production of this thing in something that is known as bioreactors. And after that, after producing them in bioreactors, I have to do some processing which is known as downstream processing. Downstream processing means separating things from everything else. Whatever protein I need, I want to separate it from everything else that is there around, like cell debris and everything. Yeah, and purification. Separation and purification comes under downstream processing. Apart from that, to preserve things, you add preservatives. For quality checking, you have some clinical trials and lot of things running around there. So this is the whole step. I'm going to explain you each of them very much nicely. Okay. So the steps and all at the end I will be explaining you, but the things that are involved, for example, restriction enzyme, what type of restriction enzymes are there, what is PCR, what is gel electrophoresis, how do you separate the DNA, all those things we have to cover first. What are the different type of vectors? Is it just the plasmid that can act as a vector, or is there something else as well? All of this thing we have to study. So we are going to study about that. So starting with construction of the first recombinant DNA. What was the first recombinant DNA? It was put down by Stanley Cohen and Herbert Boyer, my dear children. And what they did was, their objective, their aim was to link an antibiotic resistant gene with a plasmid of Salmonella typhimurium. You know, Salmonella typhimurium is something that causes typhoid, my dear children. Now, obviously, Salmonella typhimurium is a bacteria. It will be having a plasmid. What was the aim? To take out the plasmid from Salmonella typhimurium and basically put a antibiotic resistant gene over here. What do you mean by antibiotic resistant gene? That means if suppose I have put some ampicillin resistant gene over here. Suppose Ampicillin is an antibiotic, my dear children. And if I am putting ampicillin resistant gene over here, what will happen? Even if I treat this Salmonella typhimurium with ampicillin, it will still not die. Why? Because it has a gene, resistant gene over here. So this was the aim, and you might be thinking, why are we creating something like that? If we create something like that, then probably it would survive. It won't die even if we have antibiotics. So it was not like we are taking the plasmid from the Salmonella typhimurium, putting this and putting it back into Salmonella typhimurium. No, we are not going to put it back into Salmonella typhimurium. We were trying to do something else. So let's see what is that something else. I hope you understood what is a basic thing to link an antibiotic resistant gene to the plasmid of Salmonella typhimurium. So the tools that were used as restriction enzymes, which are known as molecular scissors, I told you, okay, they cut DNA at the specific sites, and apart from that, plasmid DNA, which is going to act like a vector. Okay, plasmid DNA is going to act like a vector because this is the one that transfers the foreign DNA into the host. So into the host, as in, suppose I call this plasmid DNA as a vector because it has my gene of interest. It is carrying my gene of interest. Now suppose I am going to put this plasmid of Salmonella typhimurium into E. coli. So what is E. coli over here? E. coli is the host cell, my dear children. It is the host cell. Now DNA ligase is also needed to what? To basically make them stick to each other. Now process is what? Cutting the DNA. Restriction enzyme cut the antibiotic resistant gene from a plasmid, from some plasmid, okay, from some plasmid. Suppose this was the plasmid that I was having. From here I took this particular antibiotic resistant gene. How did I cut it? With the help of restriction enzyme. And then what I am going to do is that the gene is linked to the plasmid DNA of Salmonella typhi. Again, I am going to put this particular gene over here. I am going to make a cut over here first with the same restriction enzyme, and then I am going to put the gene over here. Now what? Creating recombinant DNA. A new circular DNA molecule is formed in vitro. Yes, I am able to form in the lab. That is what it means in vitro. Now transferring to the host. The recombinant DNA is inserted into the E. coli. Now this particular thing that is there, my dear children, is put over here into the E. coli. That is the host cell. So this host cell will be now resistant to this antibiotic. Now cloning. The plasmid replicates inside the E. coli producing multiple copies. This particular plasmid, I told you, it has a site that is known as origin of replication, my dear children. You need OR. If you are a vector, if you are someone who is there to transferring the desired gene, you have to make sure that basically it has origin of replication. So this will be replicating, and that's how basically antibiotic resistant genes and everything were also being produced. Okay, that's how antibiotic resistant gene and everything was produced. So this was a simple experiment that was done. They just wanted to find out whether is it possible to create a gene like that or not. And it was done by Stanley Cohen and Herbert Boyer, and it was done in the year 1972. Works? Chalo. Now going ahead, I hope you're getting bit idea about all this thing. Now fate of alien DNA in the host, as in the alien DNA that we put over here. What is its fate in the host, that is E. coli? What happens to it? So without integration cannot multiply in progeny. With integration multiplies with the host DNA when inserted into a chromosome. So my dear children, what do I simply mean by this is, if you are making sure that it has an origin of replication, then only it will create copies. If you are inserting in a plasmid that does not have origin of replication, my dear children, it is not going to replicate. It is not going to replicate. As simple as that. Now, key factor for DNA multiplication is that requires a specific DNA sequence that is known as origin of replication, I told you, and cloning, making multiple identical copies of template DNA. That is what you mean by cloning. So, just in from this particular side, you do not need to go through very big lot of things. Just understand that you need origin of replication, that is ORI site, for increasing the copy number at all. And this process of increasing the copy number or making multiple copies is known as what? Cloning. Okay, so basically over here we saw the story of the first recombinant DNA, and then we saw what do we actually need. We need ORI site, and we have to make sure it is cloning. We need restriction enzyme, we need DNA ligase, we need a host cell, we need a vector, everything. Okay. Now, discovery of restriction enzymes. So, enzymes, restriction enzymes, as I told, is a very important thing. So, we should be knowing much things about it, right? So, discovery of restriction enzyme was done in the year 1963. 1963. Now, 1968 is not given any NCERT, but it is said the first restriction enzyme, the first identified restriction enzyme was almost around 5 years after this particular thing, after the discovery of this particular enzyme which was there. Okay, also basically not to confuse you guys, I'll just keep it, let's not confuse you guys. So, discovery of restriction enzyme basically started in the year 1963. Two enzymes were discovered in E. coli. This was the thing, okay. 2 enzymes were discovered in E. coli. One was adding methyl group to the DNA and the other was cutting the DNA. One enzyme was adding methyl group, other was cutting the DNA, and you call them restriction endonuclease. You call them restriction endonuclease because they are cutting the DNA from the middle, within the DNA only they are making the cuts. So what do I get? 1963 in the E. coli, 2 enzymes were seen. One was adding methyl group to the DNA and the other was cutting the restriction cutting the DNA, and it was known as restriction endonuclease. Now HIND2 was this first identified restriction enzyme. After 5 years they got to know that this is what HIND2, and this was done in 1968. So discovery of these 2 enzymes were found. Okay, these 2 enzymes were there, it was found, but the actual name and everything was found later after 5 years, that is in the year 1968. And the name of this particular enzyme is HIND2, my dear children, please remember it. And what does it cut? It cuts DNA specific 6 base pair sequence. That means 6 bases will be there, it is going to make cut over that site. Now it ensures precise DNA cutting at the same location every time. It does not cut everywhere, okay, it has a particular site, there only it will be cutting. So please remember HIND2 over here, it's a very, very important thing, okay. And in E. coli, 2 enzymes, one had methyl group and the other one basically was restriction endonuclease. I'll just tell you a story about this, then you might understand things better. So you know, suppose this is an E. coli. So E. coli and everything were affected by a lot of things. For example, if you talk about some bacteriophage and things like that, what does the bacteriophage do? It's a virus. It's basically inserts its genetic material over here, and it will be having its own genetic material. E. coli, it have its own genetic material. So just to make them safe, what did E. coli do? E. coli started having some restriction enzymes, started producing some restriction enzymes which had the activity to basically cut the fragments over here. So the fragments which were inserted, genetic material which was inserted by the bacteriophage was cut using restriction enzymes. But you only think it also has the DNA. This restriction enzyme can act on this also to make the whole cell understand that, hey, you do not have to make a cut over here. What are they going to do? They planned of adding what? Methyl group. So if there is a methyl group in the DNA, these restriction enzymes are not going to work over there. So this was the whole strategy. So one enzyme was there adding the methyl group, another enzyme was restriction enzymes which were basically for protection, for basically cutting down the DNA. And this name of this restriction enzyme was later found out, it was HIND2, my dear children. Okay, the first identified restriction. It's not this, but generally speaking, it's not this, but generally speaking, the first restriction enzyme to be totally found out was HIND2. And it cuts the DNA at specific sites, that is 6 base pair sequence sites. Fine. Now, current knowledge that we have about restriction enzyme is that there are 900 plus restriction enzyme isolated from 230 plus bacterial strains, and each of them recognizes different DNA sequences. Each of them have different sites, okay, and you call these sites as recognition sites. So over here, this is the 6 pair base pair sequence for them. Now going ahead, naming convention of the enzymes which are there. So for example, EcoR1 or HIND2, what do you mean by that? So EcoR1 is also a restriction enzyme, okay. Now the first letter of EcoR1, E, it stands for the genus name, that is Escherichia in E. coli. In E. coli, Escherichia coli bacteria, Escherichia E comes from this, okay, or basically in EcoR1, the E comes from the genus name, that is Escherichia. Now the next 2 letters come from the species name. For example, coli, CO comes from that, from the species name, first 2 letters of the species name. Now talking about R, this is a strain name. So represented by additional letter, that is R, from R by 13. So R by 13 was a strain. So you take the first letter, that is R over here. Now what is 1? 1 is basically put on the basis of the discovery, how it was discovered. So indicates the order of isolation, the first isolation, second enzyme, or the first enzyme from the strain, whatever it was. So it was the first strain or the first enzyme to be taken out from the E. coli, which is why its name is EcoR1. It's a very famous restriction enzyme, my dear children. Okay, so I hope you got that as well. Cool. Now please remember HIND2, 6 base pairs, and over here EcoR1 also has 6 base pairs, but this is not the first one that was identified. Okay. Now the type of restriction enzymes which are there, my dear children. So you have 2 types. One is exonucleases, another one is endonucleases. Restriction exonucleases cut the DNA at the ends. They remove the nucleotides from the ends of the DNA. Now, endonucleases, as the name suggests, endo, they are going to enter and they are going to make cut within specific sequences within the DNA itself. And what is the function of restriction endonucleases which are there? If we talk about endonucleases, they have specific function. They not randomly go and cut. They actually inspect the whole DNA from the specific recognition sequence. I told you each of the restriction enzymes which are there, they have specific sequence. The sequence for EcoR1 is GAA-TTC 5' to 3'. This is the sequence. Now, EcoR1 will be basically going through the entire DNA to find out where is this particular sequence. Now, it will bind and cut both the strands at specific points in the sugar phosphate backbone. So, please remember the restriction sites which are there, it will be always palindromic sequence. They would always have a palindromic sequence. So, over here if you see 5' GAA-TTC 3'. If you read from the opposite, the complementary end also will be 5' GAA-TTC. So, these sites are known as palindromic sites. So, restriction sites which are there, they are always palindromic, my dear children. So, whenever the enzyme comes, it is going to make a cut over here as well as in this particular strand as well. Okay, so that is why it is written both the strand at specific points in the sugar phosphate backbone. The sugar phosphate backbone, please, this pieces of information are really important. So, restriction enzymes basically inspect the DNA and cuts in the sugar phosphate backbone. This is the backbone of the DNA. Okay, now recognizes palindromic sequences, that is this. And if you talk about EcoR1, where does it go and cut? It always have the sequence that is having GAA-TTC. This is almost 6 base pairs, just like HindA2. HindA2 also has 6 base pairs, but the base pairs are not mentioned. Over here, the base pairs are clearly mentioned, and please remember 5' to 3' and 5' to 3' over here. Now, where they bind to this particular site, but where do they actually make the cut? They make a cut between G and A. Here also, between G and A, they will be making a cut. Please remember that. So, this slide we got to know two enzymes, restriction exonucleases and endonucleases. Exonucleases cuts at the end, endonucleases cuts within the DNA sequences. Okay, got it? And where do they cut? Sugar phosphate backbone. And exactly where? In the palindromic sequences, which can be read equally in both the sides. Got it? Works? Now, going ahead, the sticky end formation. So, how do you basically cut this restriction enzymes? I told you there is foreign gene and there is a vector gene. How it is cut and how it is joined and everything, how does it work? Exactly. So, let's see that. So, over here, sticky end formation. It cuts away from the center, creating a single stranded overhang. So, I told you, suppose this is the strand that we are having. Okay, this is the vector DNA. Okay, this is the plasmid DNA, suppose. The vector DNA, it has GAA-TTC site over here. Similarly, it will have GAA-TT site over here also. It is going to make a cut over here and going to make a cut over here. When it makes a cut over here, what will happen? This whole strand which is there, it will move away. This whole strand which is there, it will move away. And similarly, when you make a cut, so it's like an L-shaped thing goes off. An L-shaped thing goes off. So, if you see, there is no G over here because you made a cut over here, you made a cut over here. This L-shaped thing is being removed. Similarly, if the same EcoR1 is going to make a cut in the foreign DNA, the gene of interest, my dear children, here also it will make a cut like that. So, it is GAA-TTC, GAA-TTC. It is going to remove this particular thing again. A L shape of thing is being removed. So, from here also this is being removed. Okay, from the same side. Now, what happens? Over here you see this has no one to pair with, even this has no one to pair with. So, I will call this as sticky end. You know, the base pairs always have hydrogen bonds with each other. So, as of now they do not have a complementary pair. So, they are sticky ends. They would stick to another base pair if it is complementary. So, these sticky ends, my dear children, are made to come together and they are joined. And since they are complementary to each other, if we see T will be going with A, T will be going with A with T and A with T again. So, now it's all fine. Now, you have combined the whole DNA and this is what the recombinant DNA that you get. And still, if there are some nicks and tricks, so basically DNA ligase is the one that will be joining that. Okay. So, it creates, use in genetic engineering is that it creates recombinant DNA from different sources, that is the use of sticky ends. Now, DNA fragments cut by same restriction enzyme can be joined using DNA ligase. So, the DNA ligase is the one that is coming over here and going to make the whole process very smooth. Got it? So, I hope this is also clear. Okay, please remember EcoR1 side, 6 base pairs are there, 5' GAA-TTC, GAA-TTC, GAA-TTC. Okay, I hope that works. Okay. Now, Vectors. So we are done with restriction enzyme. Now we are going ahead with vectors, the one that carries the gene of interest. So what we understood from the whole thing is that bacteria and viruses naturally transfer genes to eukaryotic cells. They do it naturally. Okay, so you know HIV, it's in HIV, what are they doing? HIV viruses are basically putting their genetic material into us. So they have a mechanism, these retroviruses which are there, they have a mechanism to infect us. Similarly, if we talk about certain bacteria has a mechanism to infect the plants and also us as well, but it is mostly plants. For us, it is usually retroviruses that put their genetic material. In the case of plants, it is bacteria that put their genetic material into the plants. So this is a natural process. So why not use this natural process and take it for our own benefit? You didn't understand? I'm going to tell you. So basically, you know what? There is a bacteria whose name is Agrobacterium tumefaciens. Okay, Agrobacterium tumefaciens. This is a bacteria which will be having a DNA which is known as tDNA. Okay, sorry, not T. Basically, wait. First thing, it has a plasmid that is known as Ti plasmid. It has a plasmid that is known as Ti plasmid. Why Ti? It is known as tumor-inducing plasmid. Why tumor-inducing? Basically, this particular plasmid has a particular DNA which is known as transfer DNA, my dear children, tDNA. Now what will happen is that this particular Agrobacterium tumefaciens is going to infect the plants. It is going to infect the plants, and when it infects the plant, what it does is that it will transfer the particular plasmid over here. And this particular plasmid, what does it have? It has the tDNA, and this tDNA, my dear children, this particular tDNA which is there, obviously it is going to induce what? Tumor in the plants. It is going to induce tumor in the plants. So why not use this mechanism for our own need? Basically, why can't we just remove, oh my god, okay, why can't we just remove the tDNA from here and insert our gene of interest? Still it will be Ti plasmid. The plasmid is still the same. I have just removed what? The tDNA, and instead of that I have put this gene of interest. Now, this particular Agrobacterium tumefaciens does not know that I am making it a tool. This will still go and try to infect the plant, but what is it actually transferring? It is transferring my gene of interest, not the tDNA, but my gene of interest. Suppose I want the plant to be resistant to something. I want a plant to produce some toxin and things like that. Basically, I can do this. I can just do this. Okay, so basically the plant will now produce toxins and probably it won't be eaten by the herbivores which are there. So am I not doing recombination over here? Yes, I am doing recombination. And is it helpful? Yes, it is helpful. Am I not using a natural vector over here? Yes, I'm using that. So example in plants is Agrobacterium tumefaciens. It transfers tDNA into the plant cells which causes tumors. Modified Ti plasmid, my dear children, that is tumor inducing plasmid, now used as a cloning vector. It is used as a vector, non-pathogenic, to insert desired genes into the plants. Got it? Now we talk about animals. Similarly, we also have retroviruses. So naturally they transform normal cells to cancerous cells or basically some other cells. Modified retrovirus now can be used as a vector to deliver desired genes. Again, we are making sure that this retroviruses that we are using, they are disarmed. What do you mean by disarmed? Basically, they are not going to cause us any trouble. We're just putting the gene of interest and we're letting it go and attack us. Once the gene is inserted into the vector, it is transferred into a bacterial, plant or animal host where it multiplies. So whenever, for example, I am putting this thing into a vector, this is the vector, the plasmid is a vector over here. This is a vector, or probably retroviruses DNA is, or genetic material is a vector, or retrovirus itself is a vector, my dear children. Now, this particular thing, if I put it into host cell, the vector, when they are transferred into host cell, my dear children, they will be obviously replicating inside the host body. That is what is written over here. The gene is inserted into the vector, it is transferred into the bacterial, plant or animal host where it will be multiplying and increasing its number. So the only important thing that you have to remember from this slide is, in the case of plants, the natural vector that is used usually is the Agrobacterium tumefaciens, which is a bacteria. In the case of animals, always remember the retroviruses is the one that are used to basically send our DNA into the host. Cool? Once it sends it, now it does not have anything to do. So their retroviruses are acting as a vector. Cool? Works. Fine. Now, vector-less methods to make the cells competent. So there are other methods also where you do not need a vector to basically make the cells competent. What do you mean by competent? To make the cells take up the recombinant DNA. So you always remember DNA is hydrophilic, my dear children. Hydrophilic as in it loves water. But you know our plasma membrane and everything, it is towards inside it is hydrophobic, towards outside it is hydrophilic. So if you are making something that loves water go through something that hates water, can it easily pass through, my dear children? No, it becomes lot more difficult. So that is why the DNA is not usually taken up very easily. So you have to find ways to make the cells competent. Suppose this is a bacteria that I want to make competent. Okay, I want a bacteria to take up the DNA. What do I do is I have to treat with divalent cations, for example calcium chloride. Okay, I am going to treat it with divalent cations. Why? First thing, it will neutralize the negative charge on the DNA and the cell membrane. Okay, so DNA is negatively charged. Okay, DNA is negatively charged, so I have to neutralize that. So I can do it by treating with calcium chloride. Now apart from that, it also increases permeability of the bacterial cell wall. So divalent cations which are there, it makes the cell wall more permeable. The cell wall is not permeable, right? So to increase the permeability, you have what? Divalent cations. Apart from that, they also create temporary pores for DNA entry. So that is why it is known as permeable. So it is making permeable by making what? A certain temporary pores which are there. So divalent cations are there. The first thing that we do is to make the bacterial cells competent, we treat it with divalent cations, and after that we do the heat shock treatment. My dear children. What exactly happens over here is that you have to incubate the cell with the DNA on the ice. Okay, in the ice you do it, and then you briefly heat it at 42 degrees Celsius. Now it is heat. Now again you are going to cool it on ice. So this is known as heat shock treatment. This forces the bacteria to take up what is the recombinant DNA which is outside the cell. So you already made it permeable and everything. Now you are giving the heat shock treatment. Together, I will call this electroporation. When the pores are being made, I will call this as what? Electroporation, my dear children. Electroporation, followed which I give heat shock treatment. Okay, and the bacterial cells will now be taking these things up. Now what about other things? For example, I want to insert DNA into a human cell. What do I do? Without vectors, how can I insert it? Without a retrovirus, how can I insert it? Basically, there is a process that is known as micro-injection. Micro-injection, where you are going to directly inject the recombinant DNA into the nucleus of an animal cell using a micropipette which is there. Okay, into an animal cell. You can do this because animal cell does not have a cell wall. Always remember, micro injection is only done on the animal cell since it does not have a cell wall. So it can be easily inserted. Okay, to inside the nucleus, nowhere else. Inside the nucleus, the recombinant DNA has been put up. What about plants? Plants have what? Cell wall. So obviously they cannot have this particular method of micro injection, but they can undergo something else that is known as biolistics or gene gun method. What is happening over here? So you take particles of gold and tungsten, okay, specifically gold and tungsten, and you wrap it around or coat it with the DNA of interest. Whatever DNA you want to transfer, you're going to coat the DNA onto this gold and tungsten particles, and now it is shot in a very high velocity into the nucleus of the plant cell. This is known as biolistic or gene gun method. The name, please remember it, is very, very, very important. Cool. Now the third thing is disarmed pathogen vectors. So modified viruses, bacteria infect host cells and transfer. This is not vectorless method. Over here you need vector. So this is something else. So as I said, this is just one way. Okay. These all things that you saw over here, from here to here, my dear children, they all are vectorless methods. Now if you want to use vectors, you can use disarmed pathogen vectors, such as you are going to use Agrobacterium tumefaciens. Okay, you are removing the tDNA, right? So it is disarmed again. Here in the retroviruses, when you are using, again you are going to make it disarmed and only you are going to use it. So all the disarmed vectors would be coming over here in a separate category. This is a separate category. This whole thing is a separate category. So I'll just write, for avoiding confusion, this is what I'll write. Example, you have Agrobacterium and retroviruses. Cool? Works? So from here it's different. Chalo. Now features of a vector. What all features should a vector definitely have? First thing, as I said, is origin of replication. It's a starting point for DNA replication. It is the one that controls the copy number, and vectors with high copy number or ORI help recover more copies of the target DNA. More copies if you want, we should be targeting the vectors which has ORI site that has high copy number. So depending on the different ORI site, they have different types of copy number as well. It might be less, it might be more, and things like that. But anyway, origin of replication should be there because this is where the DNA polymerase would be coming and attaching and replicating the thing. Now, apart from that, it should also have selectable marker. Now, what is the use of selectable marker? It helps identify transformants, that is cells with inserted DNA. So, suppose I have over here an E. coli. Okay, I have the E. coli over here, and the E. coli will have its own things, chromosomal DNA, it will have its own plasmid, but I want to know whether it has taken the plasmid of my interest, whether it has got transformed or not. Okay, so for that I need selectable markers. It helps identify transformants, the cells with inserted DNA, and eliminates non-transformants, the cell that does not have the inserted DNA. So how do you get to know that? With the help of the selectable marker, the most famous one is the antibiotic resistant genes, my dear children. Antibiotic resistant genes. So what happens is that basically this particular plasmid will be having an antibiotic resistant genes as well. So over here it has my gene of interest, but apart from that, one antibiotic resistant gene is there which helps it survive in antibiotic medium such as ampicillin medium or whatever is there. So that's when I got to know that okay, this E. coli is able to survive. That means it has my plasmid which I was looking forward for. So this is what a transformant cell, my dear children. Suppose it is not surviving in ampicillin medium. Then the condition is like this, where there is a normal E. coli with a normal DNA and its normal plasmid. It does not have this, which is why it is not able to survive. So I am able to get to understand the difference. Why? Because it has the ampicillin resistant gene. So it is going to act like a marker. I am able to select this particular cell because it has this particular gene. So antibiotic resistant genes such as ampicillin, okay, ampicillin is an antibiotic, tetracycline, okay, chloramphenicol, kanamycin, all of them are antibiotics, and we can put their gene into our plasmid that we're looking forward for. So normal E. coli lacks the resistance to these antibiotics. The normal E. coli cells which is there, it will lack the resistance even if only if you are inserting a plasmid, a different plasmid with all these genes, antibiotic resistant genes, then only we'll come to know that. So that is a differentiating factor. You get it? So selectable markers are very much important for a vector. So this vector has it, so it is a perfect vector. Got it? Naturally, no one has. Okay? Now, third thing, it needs cloning sites as well. So cloning sites as in vectors should have few or more recognition sites. These sites as in sites where the enzymes can make cut. So suppose this is a plasmid. Okay, this is a plasmid, by dear children, and over here you see certain enzymes. This is BAMH1, this is SAL1, this is PVU1, PST1. Okay, all these are restriction enzymes, and all of them have sites over here. Suppose they do not have sites, will they be able to cut? No. Will they be able to work over here? No. So definitely you should be having each enzyme which is there, it should be having what? A single recognition site. It should not have multiple recognition sites. If it has multiple recognition sites, my dear children, what will happen? The MAMH1, suppose it have a recognition site over here and here also. It will cut the DNA over here and here also. So then the plasmid, will it have a proper structure? No, that structure will be lost. So you have to make sure that one particular enzyme has one particular site. Remember, this point is very much important. One particular enzyme has one particular site. There can be multiple enzymes with multiple sites, that's fine. But one particular enzyme which is there, suppose I am treating with BAMH1. Okay, I am treating this thing with BAMH1. So it should make a cut over here only. If it had multiple sites, it will make cut over there. But now, suppose if I have SAL1 and SAL1 again, if it has multiple sites, it will cut more site. But if it has one site, it will cut just over here. So there can be multiple enzyme site, but for one enzyme, there should be only one particular site. Works. So vectors should have few or single recognition site for restriction enzyme. So I'll reframe this. Vectors should have single recognition site for single restriction enzyme. For one restriction enzyme, it should have just one particular site. Now, too many recognition sites, multiple fragments will be happening. It will make multiple fragments and that will lead to problems. Okay. So that is the meaning of this. Now, I'll tell you a story about the selectable markers. How does it actually work over here? So see, this particular plasmid which is there, it is known as PBr322. It is known as PBr322. I'll just give you an idea about this particular plasmid, and it's really important for you to know the site for the restriction enzyme and everything. Question comes over here related to this as well. So PBr322, my dear children, it has an origin of replication for sure, and apart from that, it has an ROP site. What is this? ROP is a very important site for production of protein that is involved in replication. So ROP site is there. Now in the ROP site, my dear children, there you have a enzymes site as well. So this particular PVU2, okay, which is a restriction enzyme, it is going to make a cut over here. So PVU2 is going to make a cut in the ROP site which is there. Now going ahead, there is tetracycline resistant gene over here. This gene is responsible for making the particular organism whichever have this particular plasmid resistant to tetracycline. Okay, but there are two enzymes which have their restriction sites over here. One is BAMH1 and one is SAL1. So suppose BAMH1 cuts over here, and with this cut we are putting our gene of interest. Will it be still tetracycline resistant, my dear children? It would not be tetracycline resistant. It would not be tetracycline resistant. Please understand that. Okay. Now going ahead, this is ampicillin resistant gene. Again you have two things, PVU1 and PST1. Both of them are restriction enzyme. Either of them, if they cut over here and we put a gene of interest, this particular plasmid is not going to be ampicillin resistant anymore. Okay, but now it is. Unless and until the enzymes comes and cut, it is resistant. Now over here you have EcoR1, you have clavin site, and also you have Hind3 site. Hind3. So you should be knowing, my dear children, that what all things are there. They can ask you questions related to that as well. So if I make a cut in SAL1 site, will it be tetracycline resistant? So you will only know it if you know that SAL1 is in the tetracycline resistant gene site itself. So obviously it won't be tetracycline resistant anymore. Okay. So if I cut in PVU2 site, will ROP still work properly? So for that you should know where is it exactly, where is the site of PVU2. So please remember all the site PVU1, remember all of them are 1, 1, 1, 1, 1, 1 except PVU2 and Hind3. So PVU2 this corner, Hind3 that corner. Okay. And PVU1 is over here, PST1 over here, BAMH1, SAL1, clavin, EcoR1 and Hind3, sorry. Now you understood the basic. Now we are going to do an experiment. Okay, we are going to do an experiment with which you will understand what is the use of the selectable markers which are there. So BAMH1 site in tetracycline resistant gene is used for inserting foreign DNA. That means BAMH1 enzyme is used to cut the site over here, and it is going to insert, we are going to insert what? Our gene of interest. Now will it be tetracycline resistant, my dear children? It would not be. So recombinant plasmids lose the tetracycline resistant gene. The new plasmid that is being made, it does not have the tetracycline resistant, but retain what? Ampicillin resistant. It will be still ampicillin resistant because we are not making cut over here. Now how do we select them? So plate transformants on ampicillin containing medium. First thing that we do is we would make a medium that contains ampicillin. That contains what? Ampicillin. So what will happen is that the E. coli that have taken up the plasmid, for ampicillin, it will be able to survive. Yeah, the other ones would not be able to survive. So suppose this has taken, so I will now call this as transformants. It has taken up the plasmid over here. So this is transformant. How do I identify transformant? Basically, whatever E. coli is there, first I treated with, first I made sure that the recombinant plasmid is going or not. In some, the recombinant plasmid will go, in some, the recombinant plasmid is not going to go. Suppose this is the one where the recombinant plasmid is not going. Now I want to choose among them whether one has taken it or not. So what do? I treat this with ampicillin. Suppose a E. coli is not surviving. So that means it does not have the resistant gene, that means it does not have the recombinant plasmid. Remove it. If it has, so I got to know that this is the transformant, my dear children. So transformation has definitely taken place over here. That is cool. Now there are different scenarios. The plasmid can be like this. The plasmid can be like this, which is having both ampicillin and tetracycline. Okay, that means it is a non-recombinant plasmid. And there is a plasmid like this, which has just the ampicillin, not the tetracycline, this is a recombinant plasmid. Okay, and then the plasmid that the E. coli already has, which is not resistant to anything. So basically, now the scenario over here, so I'll change the colors a bit so you will get to understand things. Okay, works. So over here it has both, that means it is not recombinant. It is the one that does not have tetracycline, that means it is the recombinant one. And over here this is the one that does, this is normal E. coli plasmid. So this is pakka not a transformed one. Now in this scenario, my dear children, you get 3 different types of things. So E. coli with this particular thing, that is a recombinant plasmid. Then you will get another E. coli, which should I, okay, so I'll just remove this, okay. Can I remove those or should I make it here? I'll make it here. So another E. coli, okay, which is basically going to have the non-recombinant one as well. The non-recombinant one as well. So what will happen? It has both the genes for tetracycline and ampicillin. So it will also survive over here. It will also survive over here. Still it will be known as transformant. Still it will be known as transformant. But we have to find which one is a recombinant. How will we do that? For that we need another plating. We need another plating. In that what we are going to do is that we are going to add tetracycline. Tetracycline. So now here the E. coli which were there, don't worry, this is just a bit confusing, not too much. Okay. Okay. So this was the non-recombinant and this is the recombinant. How do we find out? We put in tetracycline. Okay, initially we put in ampicillin, both of them were able to survive because both of them had it. So they were transformants. At least the new plasmid got into the E. coli. So this E. coli is transformants. But I have to make sure whether it's recombinant or not. So what do? I treat it with tetracycline. In this medium, this guy is not going to survive. The recombinant one is not going to survive because it has lost the property, but the non-recombinant one will be surviving. So how do I choose? Basically, I will choose those which were able to survive over here but are not going to survive over there. So the ones that do not survive over there, they are the recombinants. I am going to choose them and I am going to basically put that particular E. coli in bioreactors and make them replicate or say increase their number. Okay, I am going to give them food and everything which is there. So that is exactly written over here. Plate transformants on ampicillin resistant medium, transfer survivors to tetracycline medium. Recombinants grow in ampicillin but not in tetracycline. Non-recombinants which are there, they will be growing in both the medium because they have both the genes of interest. Got it? So this is how it usually works. That is the use of selectable markers. But I am going to tell you one more very important way. Okay, there's a question that has come. So I am going to tell you one more very important way in which you can find out which one has the recombinant, which one is not. So that is a different way. I am going to talk about that, but wait just for a minute. So there's a question that has come. Okay, so just for you to understand that these type of questions are also framed. So ROP gene, which codes for the proteins involved in replication of the plasmid PBR322 in E. coli, is located at the restriction site of what? So I told you this type of question comes, so you know it is PVU2 2021 question. Okay, now yes, now coming to the next thing that was there, the easy method that we have. So we have another plasmid, my dear children, and the good thing about this particular plasmid is that it has a gene. It has a gene which is known as LAC-Z gene. Now LAC-Z gene you might have studied in lac-operon. LAC-Z gene is responsible for production of beta-galactosidase. Beta-galactosidase. Okay. Now beta-galactosidase, when this particular plasmid containing organism, in that particular medium itself, a chromogenic substance is added, that is a color inducing substance is added. What happens is that it goes and breaks it off. It goes and breaks it off and gives blue color. And gives blue color, my dear children. Usually this is what happens. So what I am going to do is, I am going to remove this LAC-Z gene from here. And I am going to put my gene of interest. I am going to put my gene of interest over here. Now what will happen? Now is it going to produce the beta-galactosidase? No. And when I am going to put the chromogenic substance, will it turn the color to blue? No, it will be just white in color, just white in color. What is exactly happening over here? Insertional inactivation is happening over here. Insertional inactivation. What do you mean by that? Basically, I am inserting something else, which is why the previous one is getting inactivated. Now, this is lot more easy way. Why? Basically, I just have one particular plating required. I do not need double plating like ampicillin and tetracycline. Over here, I am going to have the E. coli. With the non-recombinant plasmid, which is still coding for LAG-Z, and the recombinant plasmid, which is coding for something else. So what will happen basically when I am going to put the chromogenic substance, I am going to observe that this side is giving me blue color, whereas that side is giving me white color. So basically, who are the recombinant ones? Who are the recombinant ones? This one, because it is not going to change the color into blue. So I am going to take these E. coli, my dear children, from here. I am going to multiply it. And they are showing insertional inactivation. And this method of finding things is known as blue-white colony method. Okay, so insertional inactivation is seen in LAG-Z gene. It overcomes the need for dual plating on antibiotic plates. So that is one very good thing about what's a LAG-Z gene plasmid. Now, uses beta-galactosidase enzyme gene. Process is what? Foreign DNA is inserted into beta-galactosidase gene. This gene is now inactivated. What do you call this gene as? LAG-Z gene. Okay. Now, chromogenic substrate when added, the non-recombinants produce blue color. The non-recombinants, the one that does not have recombination, will produce blue colonies. The recombinants will produce what? White colonies. So, white colonies is equals to recombinant DNA. As simple as that. And the chromogenic substance which is being used is known as X-gal. It is known as X-gal. Works? So this is how things work. This is how selectable markers and everything are really, selectable markers are usually resistant, antibiotic resistant genes. But over here the blue white colonies and everything is also a very good way of finding things. Now insertional inactivation is happening here also. When you are inserting the gene of interest, it is not tetracycline resistant anymore. So this is also insertional inactivation. Please remember that as well. Cool. So I hope this is also done. So we were talking about the features of the vectors. It should have original replication. There should be selectable markers. So vectors, as we're talking about plasmid and everything. So the plasmids which are there, it should be having some certain selectable markers. It becomes easy for us to understand what is recombinant and what is not. So transformants are the one that take up the plasmid, any new plasmid. Recombinants are the one that have taken up the recombinant plasmid which is there. Okay, now apart from that, cloning sites or recognition sites, basically for one enzyme there should be only one site, otherwise it will lead to multiple fragment formation. And this is how I explained how the selectable markers work, and then we spoke about the blue white colony thing as well. I hope this is clear. Okay, done. So we spoke about vectors, we spoke about vectorless methods, we spoke about features of vectors, and then also about restriction enzyme, the type of restriction enzymes and everything. There's a question. An antibiotic resistant gene in a vector usually helps in the selection of. An antibiotic resistant gene in a vector usually helps in the selection of what? Non-recombinant cells, competent cells, non-competent cells, transformed cells. So it will be transformed cells. I would be able to understand whether it is transformed or not, because usually the cells do not have antibiotic resistant gene. We are going to put it. That's how we are transforming it. Competent cells as in the cell that has the capacity to take up things. So for that, there's not a method. It is transformed cell. Cool. Now next question is not there. Or did I have the question? But next, you have Abhyas KCET test series and Lakshya KCET crash course, my dear children. So you see, you might have understood, at least I'm not sure if you understood a lot of things, because this chapter is a little confusing on one end, but I tried my level best trying to make you understand. But my dear children, practice would make this thing better, more than anything. So basically you have to keep on practicing things. How you can practice is with the help of test series, my dear children. In our Abhyas test series, we have certified full syllabus and chapter wise tests available. There will be detailed video solutions, there will be one-shot revision videos. We have done past 5-year paper analysis, and this is all for ₹1499. Apart from that, if you think that you do not want a test series, but basically apart from that, I also want a crash course, I want to study come to the campus and study. For that we have Lakshya KCET crash course, my dear children. There will be daily practice tests, one-on-one doubt sessions, guided study hours, PYQ focus, everything. Full syllabus coverage will be there. It's a 21-day course. It's there in Bangalore at our campuses. So there will be separate teachers for teaching you who have a legacy of bringing out what? KCET rank holders. So if you have any doubts related to that, my dear children, you can put a call to this particular number. And also the link might be given in the description box. You can just check it out. Going ahead, so over here, so basically we did all that. Okay, what we did, we spoke about vectors, restriction enzymes and things like that. I told you in the initial part of the chapter, okay, when I started off this particular chapter, that after cutting the DNA which is there, you know, you get so many fragments, so you have to separate them, and you do that with gel electrophoresis. So let's talk about that also. Let's talk a bit about that, otherwise it will be a big problem. Right, where is it? Where is it? Where is it? Where is it? Yeah, it's there. So separation of DNA happens like this. So DNA fragments are produced by restriction endonucleases, and this method is known as gel electrophoresis. I'll tell you something. So there is cathode at one end. So this is the cathode end, and there will be anode at one end. Okay. Now anode is positively charged, cathode is negatively charged. Anion is negatively charged and cation is positively charged, but here it's opposite. Okay, anode is positively charged and cathode is negatively charged. DNA, what charge does it have? It has negative charge. It has negative charge, my dear children. So over here, if cathode is negative charge and we are putting DNA at this particular wells which are there, in this particular wells which is there, we are going to put the DNA. And what is over here? It is a gel. It is agarose gel, my dear children, which you obtain from the seaweed, Now what is going to happen is that the DNA fragments, they see it's a negative end over here. I have to basically repel, I have to move away from here. So it will move away to the positive side. And the one that are smaller in size, they will be moving really fast. The one that are larger, they will be moving really slow. Always remember, the DNA which is going to move is going to sieve and move. It is moving from within. It's not like on the surface of the gel it is moving. It is having a sieving effect. This gel which is used, it has a sieving effect. It has a sieving matrix through which the DNA will sieve and move. Please remember that, very important thing. Now the DNA has moved like this. Basically, how do I view this? How do I understand where the DNA and everything? I have to take the help of one particular stain. And what is this stain? It is known as ethidium bromide. This is a very important question. What do we have, dear children? Ethidium bromide. Now ethidium bromide, again, it won't be visible unless and until you are putting UV light. So when you put UV light, what will happen? The ethidium bromide will give a bright orange bands. That's when it becomes visible. And now you want to extract the DNA. You have to remove the DNA from the gel, right? What do you call this process as? You call this process as elution. Now elution is given like this in your NCERT and you tend to not take care of it. You not basically take it into consideration. But elution is a very important process. What is it? Elution means you are extracting the DNA from the gel, not from anywhere else, just from the gel. When you are extracting the DNA, it is known as elution. Now the purified DNA fragments are used in recombinant DNA construction. So this, after that, it is purified and basically then it is put in the plasmid and everything for making a recombinant DNA. I hope you got it. Okay, so please, what you have to remember is that DNA is negatively charged. It will move towards the positive end. The smaller ones will be moving really fast and it is going to sieve and move. It is not going to move on the surface, but it is going to sieve and move. All these little things are really important. Now, to visualize this thing, what do you need? You need UV light, my dear children. But before that, you have to stain. What do you stain it with? Ethidium bromide, EB, ethidium bromide. Okay, once you stain with ethidium bromide and put UV light, it will show bright orange color bands. Now, you have to remove the DNA. The process is known as elution. With the help of elution, you can remove the DNA very much easily. Extract the DNA. Got it? So agarose gel from seaweeds acts as a sieving matrix. I hope this works. So smaller fragments move farther than larger ones. Okay. Now we will talk about the elaborate explanation of each of the points. So first we spoke about the points in general, then we spoke about the tools that are required. You need vector, you need restriction enzymes, and you need the electrophoresis to basically separate and things like that. Now I am going to explain it one by one detail. Okay, first thing, we saw isolation of the genetic material. I told you in my from my whole genetic material I want my DNA and all. How do I basically separate it? So first thing, DNA is a genetic material in all organisms and must be in pure form, free from RNA, proteins, polysaccharides and lipids, that is RNA, proteins, what do you call it, carbohydrates and the lipids, that is fats, for restriction enzyme action. Okay, so what do we do is that, first thing, there is cell, there is what? Cell. Inside the cell there is nucleus, inside the nucleus there is genetic material. So in some organism you have an extra layer that is known as cell wall, such as plants and such as fungi. Okay, some plants and fungi basically they have what? They have and also bacteria. Bacteria has it made up of peptidoglycan. So basically cell wall is there. So cell lysis has to be done first. So you need enzymes to break the cell wall. Now suppose in plants the cell wall is made up of cellulose. In plants the cell wall is made up of cellulose. So what enzyme do you need? You need cellulase. You need what? Cellulase. Now in the case of fungi it is made up of chitin. It is made up of chitin. So what enzyme do you need? Chitinase. In the case of bacteria it is made up of peptidoglycan. Okay, it is made up of peptidoglycan. So what enzyme do you need? You need lysozyme. And I'll tell you, lysozyme is there in our tears. In our tears there is an enzyme lysozyme, which is why we tear up if something comes in. Okay, in the saliva it's there. So it is a natural mechanism of breaking down the bacterial cells which are going to going to attack our body. Okay, I hope you can remember by learning like that. So this is the first thing. So you should know, if you want to isolate the DNA from a plant or a fungi or a bacteria, you need these many enzymes. Now what? Removal of macromolecules such as RNA. Suppose you got in. Now you will be having RNA and everything. So RNA, how can you remove it? You can remove it by using ribonuclease. Okay, ribonuclease or RNase. So DNA is cut DNA, RNase will be cutting the RNA. So ribonuclease can be used for basically cleaving the RNA. Now protein can be removed by using protease. Protease are going to break down the proteins. Okay, now other molecules are removed by appropriate treatments. For example, lipids are broken down by lipase. Okay, polysaccharides can be broken down by amylase and things like that. So there are so many enzymes around it. Okay, so RNase for RNA, protease for proteins, okay, lipase for lipids and things like that. Once, please do not use DNase. If you are using DNase, it is going to cut the DNA. You want the DNA to be intact. So please don't treat with that. Now, once you have treated with all of this thing, what you are going to do is you are allowing the DNA to precipitate. How you can do that is that you should be adding chilled ethanol. When you are going to add chilled ethanol, the DNA is going to precipitate like fine threads in a suspension, which you can see over here. So the DNA which was there, it is going to separate out like this when you put chilled ethanol. So children usually put ethidium bromide over here. Ethidium bromide is a stain, baba, and chilled ethanol over here is just added for making the DNA precipitate out. And this process where you are going to take it by outside is known as spooling. It is known as spooling. Fine. So these are things that you have to know. These enzymes are really important. I'll show you a question that have come up. Okay. So from the following tools, techniques of genetic engineering, identify those which are required for cloning a bacterial gene in animal cells. You're putting a bacterial gene in the animal cell. Choose the correct option. So what all do you need over here? What all do you need over here? My dear children, please remember, it's an animal cell that you are going to insert things in. It's a whole says animal cell. So if it is animal cell, obviously you need retroviruses. Is retrovirus mentioned over here? No. Then that is not vector. You are not using a vector for that. You are using vectorless method. So if it is a vectorless method for animal cells, what do you need? You need micro-injection. They cannot undergo gene gun method. So this is wrong. Okay, this is wrong, entirely wrong, right? And now what? And now what basically, my dear children? Tell me, can we use agrobacterium timifictions again? No. We are not going to use that as well. We need what? We need either retroviruses or basically we can use micro-injection. Okay, now going ahead. Do we need endonuclease? Yes, because you have to cut. You have to cut. So endonuclease is required. Do you need lysozyme? Do you need lysozyme? Actually, it's not lysosome, it's lysozyme, which are required for cloning a bacterial gene in animal cells. Lysozyme required or not? You tell me. Lysozyme required or not? Bacha, from the bacterial gene, that means you have to take it from bacteria. So for breaking down bacterial cell, what do you need? You need lysozyme. So this is required. This is definitely required. Do you need cellulase? No. Cellulase is in plant cells. You do not require that. Electrophoresis, is it needed over here? Yes, you need it. You have to separate it, right? So this is also needed. Ligase, need it over here. Yes, you need it. So all these things you need, but not 1, 2, 3. You don't need these 3 things. So I'll say 1 you need, okay, 1 you need, and then you need 2. You need 2, and then you need 6. 1, 2, 6. Where is it? 1. Sorry, not 6. 1, 2, and 4. 1, 2, 4, and then this lysozyme also you need, that is not mentioned over here, and then you need 8. So I'll say this is the correct option. Rest all option has 3 in it. Rest all option has 3 in it. So I do not need the options with 3. I do not need the options with 3. Got it? I hope you got it. So this type of questions can come, which can be a kind of tricky. Even I got tricked because as of now I am not in a zone to solve the questions, okay? But still I'm telling you, these type of questions can come and you have to be really careful. What are the terms given over here? So bacterial gene, as in you have to take the cells, things from the bacteria. So for breaking down the bacterial cell, what do you have? You have lysozyme. You have lysozyme, okay, not cellulase. Cellulase is needed for a plant cell. Okay, now you're putting an animal cell, that means you need micro-injection, not agrobacterium tumor fissions. Okay, or if you're using a vector, then it should be retroviruses. Endonuclease definitely you require for cutting. Okay, ligases you need for sticking, electrophoresis for separation. Rest everything is correct. Okay, I hope you got it. This is a 2023 question. So these type of questions also come. Please be prepared for that. So if you're not aware about all the concepts that we studied, you won't be able to answer this. Not to scare you, but just to make you aware that you have to practice the questions very much, my dear children, and we are going to do that really soon. Okay, if you're taking the test series, please practice the test series really well. It is going to be really helpful. Apart from that, we are also going to come up with some live sections and things like that for PYQ practice. Yeah. Now, a student while extracting DNA from Aspergillus fungus enzyme to break open the cell wall is. So Aspergillus is a fungus that is mentioned over here. So the enzyme that is needed to break down the cell wall of fungus is what? Chitinase. Over here, chitin is a thing that is there in the cell wall of the fungus. So if you do not know chitin is a thing that is a component of the cell wall of fungus, you would not be able to answer this. But my children know everything because we have studied it together. Haven't we? Haven't we? We have studied it together over here, right? So if you are very much sure about this particular thing and also the vectors and everything, the chapter becomes a lot more easy. And I think you have already completed halfway, not halfway, it's 3/4 is done. Now a little more is left and you are doing a wonderful job. So please do not get disheartened. You're going to ace all the exams. This is how every time you study something new, obviously you will feel that I am not going to do well, I'm not going to do well. Now you might have studied for PU2. The thing is quite different for KCET as well because that is a competitive exam, right? It's a competitive exam, so it will have its own differences, but it's not difficult at all. You can do it, trust me on that. Okay, so this is the isolation. First is isolation. After isolation, we are going to do what? We are going to use restriction enzyme to digest it. You know, I already told you, you basically take the DNA, cut it with restriction enzyme, and then you run in through gel electrophoresis. So restriction enzyme digestion is done, then you basically separate with the help of gel electrophoresis, which is mentioned over here as well. So this is the same point. Yes? Yeah. So the DNA is negatively charged, it will move towards the positive electrode, that is the anode. So restriction and digestion, separation of the DNA. Now what? Amplification. Now this is something that we didn't study. So amplification is basically when we have the DNA, the smaller fragments, now we want to increase its number. So what do we do is that the DNA which is there, first thing we know is denaturation. We want to separate the DNA. That's when a new replication can happen, right? So first step that is there, it is known as denaturation, my dear children. So it looks something like this. So your DNA strand was like this. So I separated them into 2 other strands. Now what? I want to have DNA replication to happen. If I want DNA replication to happen, I have to make sure that primers are attached. Because you know what? DNA polymerase, which is responsible replication, cannot start off the replication by itself. It needs certain primers. So this process of putting the primers, my dear children, it is known as annealing. It is known as annealing. And DNA polymerase works from 5' to 3' direction. So the opposite strand should be 3' over here. That's when the primer will come and attach over here and will move. So the template strand which is there, always remember it should be 3' to 5'. The DNA polymerase will make a new strand from 5' to 3' direction. So primer is attached over here and it is attached over here as well. This process is known as annealing. This process is known as annealing. Now you are going to take the DNA polymerase, and here the name of the DNA polymerase is known as Taq polymerase. Why Taq polymerase? Because it is excluded or basically extracted from Thermus aquaticus, which is a bacteria. Thermus aquaticus is a bacteria that can survive high temperature, and you know what, in this particular technique, that is PCR, high temperature is being used. So obviously the DNA polymerase has to survive over here, which is why we extracted from Thermus aquaticus, a thermostable bacteria, my dear children. Okay, now apart from that, you also need deoxynucleotides. Okay, you need substrate, right? All of this, what are you going to add over here? So Taq polymerase and also deoxynucleotides are needed over here, and now it is going to extend, and this process is known as extension. This process is known as extension, and it can undergo many cycles to amplify the DNA. So first thing, components required is what? Template DNA. You need the template DNA, yeah. Now two primers, you need two primers over here to attach, and DNA polymerase, yeah. And this DNA polymerase, it should be thermostable enzyme. It should be an enzyme that can survive at high temperature. Now deoxynucleotides, the building blocks of DNA, also you need that for extension, right? Now the process, denaturation happens in high temperature, that is 95 degrees Celsius, almost at 95 degrees. This is very important. These temperature details are also really important. So it separates the DNA strands at this particular high temperature. Okay. Now annealing, the primers bind to complementary sequence. The primers which are there, it will bind over here, and this will happen almost around 50 to 65 degrees Celsius. It's actually 50 to 55 degrees Celsius, not 65, 55, but yeah, you can just go through. Now extension is DNA polymerase. Basically when the Taq polymerase from Thermus aquaticus synthesizes the new strand, it happens in 72 degrees Celsius. If you see, the temperature is really high. Okay, now repeated cycles are required. The cycle repeats multiple times, amplifying the target DNA up to billion copies. Now the amplified DNA can be ligated into the vectors and can be put for cloning. I hope you got it. Okay, works. Now one very important thing, there's a question that if you have little amount of pathogen and if you want to increase the, what do you call, the DNA or the genetic material, what method do you apply? This PCR only, polymerase chain reaction. It is the method to amplify the DNA in the lab. Please remember that. So even small sample sizes there, we can increase with the help of DNA replication, polymerase chain reaction. Okay, so amplification is also done. So you isolate it, you cut with the help of restriction enzyme, then you ran through the electrophoresis changes, you separated it. Now you took the separated strand, you amplified it. Now what are you going to do? You are going to transfer the DNA. Okay, first thing, you are going to put the DNA into the plasmid, and then you are going to transfer the recombinant DNA into the host cells, which is there. So first we have to make the recombinant plasmid, and then we do this. Okay, so there will be a fourth step which involves what? I'll put it over here so that you don't get confused. Making of recombinant DNA and then transfer of recombinant DNA into the host cell. So competent cells take up recombinant DNA from the surroundings. The competent cells, as in the cells which are capable of taking in, it will be taking up from the surroundings. Example, to understand the transformed cells grow in ampicillin medium, the cells survive. All of this we have discussed previously as well. Just give me a second, okay? Yeah. So basically we discussed about this, how to understand what is recombinant, what is not. That technique is being done. Okay, basically the one that grows in ampicillin, that is again put in tetracycline. If it is not growing in tetracycline, that means it is recombinant. All these things we have already discussed. Just know the method. Okay, now after that, once that is all done, we take the recombinant ones and we are going to see whether recombinant ones are going to produce what? The protein that we are looking for. So expression of the foreign gene. So after cloning, the foreign gene must be expressed to produce the desired protein. So this protein is being produced by this particular organism or not, that is being checked. And once it is being checked, okay, first it has been sure that it has the recombinant DNA, then the expression is being checked. If it is all good, what we are going to do is that we are going to take this organism, the bacteria and all, we are going to put it in a bioreactor, and this will help in the large scale production of the proteins, because the bacteria are going to basically feed on the food that we are going to provide them, and they increase the number. When they increase in number, they are also going to clone the plasmid that we have put in them, and the proteins also increases. Okay, so continuous culture system is being there, fresh medium is added, and used medium, whichever is there, it will be removed because it will have so much waste and that could be toxic, right? Now, keep cells in active exponential growth. Always remember, you know, there are two types of growth. One is exponential and the other one is logarithmic growth. Exponential means it keeps on increasing, the growth keeps on increasing. But it only happens if the resources are not limiting. If the resources are not limiting, that's when it will grow like that. In nature, in our condition, okay, do we have exponential growth? No. There's a logistic growth. Why there's a logistic growth? Because the resources are limiting, my dear children. But when you're talking about bioreactors, we are making sure it shows exponential growth because we are providing it foods. So there is no limiting resource. Always remember, in bioreactors, active exponential growth is being seen. Now, bioreactors for large scale production processes large volumes, that is 100 to 1000 liters of the culture. They convert raw materials into proteins, enzymes and other products, provide optimal conditions, that is temperature is being seen, pH is being controlled, oxygen, nutrition, everything is being controlled over there. Okay. Now, stirred tank bioreactor, which is this, my dear children, it has cylindrical shape with curved base, okay, that allows in better mixing. There's an agitator system. What does agitator system do? Helps in even distribution of all the things. Now, oxygen delivery system is also there so that the oxygen could be delivered easily and it can be given to all the different parts, okay, of this particular whole thing. Now, foam, temperature and pH control system is there, and sampling ports are also there so that we can take out the sample and test for any toxic thing or things like that. Okay, so this is a stirred tank bioreactor. If you are asked for the picture, just understand this is stirred tank. There is one more, that is sparged bioreactor, where actually in sparged, no, the oxygen delivery is much more better than stirred tank bioreactors. Okay, just have your understanding regarding that, but that is not usually being asked. So, but you should know it is stirred tank and sparged bioreactors. In sparged, the oxygen delivery is much more better, but yes, here also things work. Cool. Now, so you are increasing its number with the help of bioreactor. Now, what do? After you're done with the whole process, you are going to do downstream processing. What do you mean by downstream processing? Basically, you want the protein. You want to take that protein. So, you have to separate things down and you want to purify it. You have to make sure that it's just a protein that is going to come, not anything else. So, this process which involves separation and purification, okay, separation and purification is known as downstream processing. After which, okay, after doing this, preservatives also can be added to preserve things. Okay, it can be added for preserving things, but please remember in downstream processing you have two things, separation and purification. And then after that, as I said, formulation is where you add preservatives and everything. After that, quality checking is being done, clinical trials are being done, rigorous testing to meet the standards, products are, so processing steps vary depending on the different product. That is just given, that is an extra point, you don't have to go through it just to make you confused. Okay, so quality control is being done, clinical trials are being done, everything is being done, and later this product is being released to the market, whichever is there. So, you know what, in our body there is alpha-1 antitrypsin. You might have heard about it in the next chapter, that is biotechnology and applications, you study about it, right? So, I think in that chapter only, right? Yeah. So, alpha-1 antitrypsin is a protein that is used to treat emphysema, that is a respiratory disorder. It's a human protein, but bacteria are basically made to be recombinants to produce this. So, they are produced and that particular thing is being used for treatment of emphysema. So, these type of things are being produced with the help of the technique that we just studied today. Okay. So, I hope you remember all the information, separation, purification, please remember it, and apart from that, all this thing. And with this, we are completing the chapter. So, I know this might have been a bit tricky here and there to understand, okay, but if you go through this once in your NCR, from your NCRT as well, now you will understand it lot better. Everything will just sink in. So, if you have time, go through it. Otherwise, just start doing questions. If you are someone who thinks that, okay, ma'am, I think I should have one read from NCRT also, then only I will understand it, go read it. If you are someone who totally understood it, first thing, please like, share and subscribe, and also comment. And one more thing is that if you are not understood, then as I said, have your NCRT. If you have understood, just do lots and lots of previous year questions which are there. And if you're finding difficulty in doing certain questions, you can put down in the comment section. I will help you out with the questions. Okay, put down the whole question whichever is there. So, I'll try to answer it from my end if at all you're not sure about it. Cool. works. So I hope it was explanatory and by itself, and my dear children, you'll get the PDF and everything in the WhatsApp group. Do not be worried. Wishing you all the best, okay, all the best, and stay blessed, and bye bye, take care.